Good day everyone, I am J.I. Hanayro from De La Salle University and a regular member of the Chemical Sciences Division of the NRCP. And I will be discussing the role of chemicals in mediating communication in plants and insects. The topics in this lecture are divided into three. The first topic introduces semiochemicals or the chemical signals used in communication. The second topic provides a general overview of gas chromatography mass spectrometry or GCMS, a common technique used for the study of semiochemistry. Lastly, the third topic presents some practical applications or consumer products that are developed with the help of semiochemistry. By the end of this lecture, you should be able to describe the different semiochemicals and the mechanism in which they elicit physiological response, discuss the basic principles of GCMS, and finally, explain the role of semiochemistry behind some of the common products that we use. Are you ready? Let's go! Aren't you amazed that we could understand each other? Apart from the words that I speak, my actions and expressions can also be used to convey information. How about plants and insects? What signals do they use to communicate with each other? How do insects tell their partners that it is time to reproduce? Plants and insects speak the beautiful language of chemistry, wherein they release specific chemicals as a signal for a particular event. When a group of insects is threatened, they release chemicals to alert each other. When it is time to mate, insects also release specific chemicals to attract their partners. When insects are hungry, they follow particular chemicals emitted by the plant that they will eat. The release and recognition of these chemical cues is central in their ability to communicate. In most cases, these chemical signals are low molecular weight and volatile. This means that these chemical cues are quickly evaporated and can easily be diffused in the air. This class of signaling compounds are commonly referred to as volatile organic compounds or VOCs, and oftentimes they bear a distinct smell or odor. You might wonder, why do insects use chemicals as a communication tool? While a definitive answer is difficult to attain, one plausible answer is due to the diversity of compounds available. To further drive this point, can you imagine the smell of a banana? How about the scent of a pear? Believe it or not, the chemicals responsible for the odor of these two fruits only differ by one methyl group. The chemical structures are very similar, yet the physiological response elicited from us is very distinct and unique. These signal chemicals are generally called semiochemicals, derived from the Greek word semeion, which means to signal. When a signal chemical acts on the same species, we call this particular type of semiochemical as a pheromone. The action of pheromones can include inducing for mating, alarm, aggregation, among others. If the semiochemical mediates interaction between two different species, this chemical is referred to as allelochemicals. Allomones are often released for defensive purposes, while pheromones are often emitted by plants and are used by insects to locate them for feeding. A good example of cynomones are floral scents that attract pollinators. For anemones, a classic example is Limburger cheese, which attracts the mosquito, Anopheles gambiae. How do insects process this chemical information? Central to this molecular recognition process are odorant binding proteins or OBPs. These proteins are abundant in the antenna of the insects, where scents or chemical signals bind to them. The binding of odors to OBP is essential in order to enhance the solubility of these compounds in a polar environment. The OBPs then mediate the transfer of the odors to the appropriate transmembrane receptors, which will trigger the G protein pathway. This will further activate several pathways such as voltage-gated channels, which will lead to the regulation on the influx or efflux of ions, ultimately leading to the perturbation of the membrane potential, which will serve as the basis of the insect's physiological response. Aren't semiochemicals amazing? If we can decode the chemical messages that are released and recognized by insects, we may be able to use them for our advantage, such as for pest management. But how can we study semiochemicals? How can we discover more of them? Semiochemicals, as discussed earlier, are mostly volatile organic compounds. And to identify these semiochemicals, analytical techniques have been developed to collect, separate, and determine its identity. To collect these volatiles from a specimen, for example a leaf, one extraction method is the use of solid-phase microextraction fiber, or SPME. It looks like an injection wherein the needle part is a long and very thin fiber made of an absorbent polymer. Collection of the VOCs is very straightforward. You just have to place the sample in a sealed container, allow it to equilibrate, and have the needle exposed in the headspace region or the top region for a few minutes to allow the VOCs to adhere to the fiber. And you're done! Collection complete. After this, the needle fiber is then injected into your GCMS. It is only by exposing the needle fiber to high temperatures that the collected VOCs detach from the needle fiber. We call this fiber desorption. The GCMS is an analytical instrument that separates the components of a mixture based on differences on their boiling points and identifies the identity of the unknown components. It has two parts, the GC or the gas chromatograph and the MS or the mass spectrometer. Say you have a mixture composed of acetone, limonene, dodecaine, and an unknown component. The GCMS is highly capable of identifying this unknown component in qualitative and quantitative analysis. It starts by first injecting the mixture in the injector port of the GCMS, where the molecules get released and then travel to the GC column. In the heated column is where the separation happens based on differences on boiling point. However, other factors such as the column's dimension, thickness, length, carrier gas, and chemical phase properties also influence chemical separation. After the GC, the separated mixture components travel to the MS system. In the MS system, it passes through the ion source where ionization happens and the compounds get fragmented. A particular compound fragments in a distinct manner, and these compound fragments eventually get detected, measured, and identified. By studying the structure and mass of the fragments, you can determine the identity of the compounds in your sample. It's like a chemical puzzle. Alternatively, modern GCMS systems have a huge library of compounds where the computer can scan the library and tries to match the fragmentation pattern of the compounds from your sample. Now that we know what are semiochemicals and we also know how we can study them, let us go through some commercial products that use semiochemicals or disrupt semiochemical sensing. Let us explore the practical applications of this class of compounds. One great example of semiochemical disruption is when we use mosquito repellent products to fend off those blood-hungry mosquitoes from us. Mosquitoes have poor vision, so they rely on their sense of smell using receptors in the tiny hairs located on their antenna to locate food, good spots for laying their eggs, and to find their next human prey to feed on blood. These receptors can smell certain chemicals from human odors such as lactic acid from sweat, carbon dioxide from our breath, and they can even sense body heat. They also have taste receptors located on their feet, so they know that they are in for a treat when they land on food or on a prey. Long before until now, natural plant extracts like citronella and eucalyptus oil were used as a repellent. They are effective but they wear off faster. Scientists have then extensively searched for a compound that are longer lasting and would stop them from biting humans to spread diseases. They then developed compounds such as picaridin and diethyltoluamide, or simply known as DEET. You should probably see either one of these compounds present in common insect repellent lotions. These compounds may hide or decrease the volatiles coming off from humans, and a combination of these repellents and human odors would present a different overall chemical profile to the mosquitoes that the human prey would render invisible to them. If that isn't enough, remember that they can get a taste of you when they land on you, and these compounds really taste gross for them to eventually leave you alone immediately. Ants are another example. Don't you hate it when you leave food on the table for a second and you come back and see a bunch of ants devouring your unfinished dessert? Ants leave a trail of pheromones that belong to a class of compounds called pyrazine that serve as communication for other ants to follow. Ants use their antenna to detect these pheromones. The ants release more of these pheromones as the perceived value of the food resource increases, attracting more ants to utilize those valuable resources. A usual but temporary way to stop the trail is by drawing a chalk line to break the pheromone trail. You will notice that the ants would stop, look confused, trying to look for the trail. Usually this is only temporary and they are smart enough to get back on track. There are also household products that utilize these pheromone trails to backfire on them by using attract and kill traps to efficiently eliminate insect pests such as cockroaches. Cockroaches are difficult to extinguish because they reproduce exponentially. They love to feast on stale food and crumbs lying around our homes. These baits are in the form of cockroach gels and are usually made of protein-based ingredients meant to be applied in dark places and corners in our homes. These discrete baits may contain semiochemicals from food sources that lure the roaches to think that there are food sources nearby. These are found to be 1-hexanol, which is a volatile emitted by decomposing lipids, ethanol from fermentation of foods, and DDMP, which is the scent of a caramel-like flavor present in foods. These gels may contain borax, which upon ingestion of the roaches, may lead to the impairment of their digestive system. This will render them unable to feed, which will eventually lead to their demise. In our lab, we also study semiochemistry and try to develop pest control products based on them. In our ongoing project funded by DOST Picard, we discovered that the compound caryophyllene is a pheromone for the cacao meridbug or CMB, Helopeltis bacceri poppius. The CMB is an emerging pest for cacao wherein it uses its long proboscis to puncture cacao pods for nourishment. This results to the rotting of the punctured cacao pods, rendering them useless. Common pest control strategies employed in cacao farming involve the application of pesticides and covering the cacao pods with plastic bag to protect them from pests. Thus, the utilization of semiochemicals for pest management offers a more environment-friendly way of controlling these pests. We utilize GCMS to characterize the compounds emitted by the feeding hosts of CMB. We then tested the compounds to evaluate the response of the CMB using a tool called olfactometer. As you can see in this image, most of the CMBs were attracted to the chamber that contains the caryophyllene compared to water. Currently, we are designing traps to incorporate caryophyllene as the CMB lure. Apart from pheromone, we are also working on the elucidation of the pheromones for CMB. For this research, we use GCMS to characterize the compounds emitted by sexually mature CMBs and compare their chemical profile with juvenile CMBs. Through extensive comparison, we were able to detect several compounds that were exclusively observed in the mature CMBs. When we exposed male CMBs to this compound, we observed that they were attracted to the compound, as well as their behavior significantly changed. In particular, the male CMB started to mount another male CMB, thinking it is another female because they can detect the pheromone. As you can deduce from this, pheromones can also be used for pest management since it does not only attract or lure in the insects, but can also disrupt their mating, feeding, and other behavior essential for their survival. We have now reached the end of this lecture. Congratulations for completing it. To summarize, you learn about semiochemicals, which are signal chemicals used by plants and insects to communicate with each other and with their surroundings. There are different kinds of semiochemicals such as pheromones, cheromones, cinnamones, among others. These semiochemicals are recognized by insects through odorant-binding proteins, which are abundantly expressed in the insect antenna. Considering that most semiochemicals are volatile, studying them requires the use of a gas chromatography mass spectrometer. A GCMS analyzes and separates volatile compounds based on their boiling points, and the mass spectrometer is responsible for fragmenting the compounds in order to uncover their identity. Finally, you are now aware that some of the consumer products that you use, such as mosquito-repelling lotions, have been developed thanks to semiochemistry. In addition, semiochemistry also makes it possible to protect our crops, such as cacao, against infestation. Thank you!